So the next level of biodiversity or type of biodiversity is genetic diversity. And this is the genetic variation within species, both among populations in space and among individuals within a single population. So why is genetic diversity important? So sit and think about for a second, what does genetic diversity do for us? What does it do for organisms? One is it allows species to persist into the future. So you can't evolve or adapt as a species without genetic diversity. It's necessary for adaptation and evolution to occur. And the environment is predicted to change. It's always been changing. And so all species need genetic diversity in order to adapt with their changing environment. So it's key for the future evolution of a species. We don't want to just conserve a static species. We want to conserve their ability to continue to change with the environment. It can also protect species from harmful genes. So if there are more genes in a population, if a few individuals have some harmful genes that mean that they don't survive well or that they don't reproduce well or that they produce offspring that have some kind of problem, then that species is going to have diversity so that not all of the individuals have that. If all the individuals have the same genes and there's a harmful gene that impairs survival or reproduction, then that's going to be problematic if we want the species to persist for a very long time. Genetic diversity is also very important for people. Some of our crops don't have a lot of genetic diversity. The classic example is the banana. All the bananas in the stores are a particular variety of banana called the Cavendish banana, and there's very little genetic variability. And they're really concerned about a disease coming along that we just don't have genes to put back into the bananas to help them be resistant to a particular pathogen. And so that can be really helpful for our crops to have genetic diversity. So we can breed and make new varieties of different crops so that they're more drought resistant or that they have more nutrition or that they're resistant to some kind of pest that is becoming more abundant. Things like that. And that's both important for crops and domesticated animals. Genetic diversity is also really important for drug development. So genes that code for unique compounds that we can make into pharmaceuticals. And then there are also just inventions, not necessarily just drugs, but all kinds of different properties from species help us make the new glue or design new different technologies. And those genes and that genetic diversity can help us have these things available if we want to develop these new inventions. So an example of why genetic diversity can be really helpful in the face of environmental change, this one has to deal with cane toads, which toads are not native to Australia at all. And so cane toads were introduced to protect sugar crops from the sugar worm. And so they introduced these toads with the idea that they would control this sugar worm and we'd get better sugar production in areas in Australia. Unfortunately, the cane toads don't eat those cane worms at all, and they don't help with sugar production. But yet now there are tons of these cane toads all over Australia. And all toads have a poison gland, and you can see right here, this gland right here on the cane toad has lots of poison. And there are no toads native to Australia, so this is kind of a new thing for different predators to deal with. But the poison is really potent. And so lots of predators have had really big problems. They bite down on this toad that doesn't move very fast. They think they found this easy prey. They get a mouthful of poison and they die. So there was a particular kind of snake that they studied, the red-bellied black snakes, and they've actually evolved smaller heads, so they're not able to eat this very large toad, and that has increased their probability of survival because they're not getting poisoned by the toads by trying to eat them. And so this is an example of if there hadn't been this genetic variability for head size in the population of snakes, then they would not have been able to adapt to this new introduced potential prey item that's actually really poisonous. Just to talk quickly about some of the terms we use when talking about genetic diversity. So genes are the physical units of heredity. They were originally talked about by Gregor Mendel. So if you remember the guy that crossed the pea plants, he's the one that figured out about genes. And now we know that there are these defined DNA sequences. So genes are the sequence of genes that code for something. And they're heritable because our parents pass genes on to us, and then we will pass them on to our children. Chromosomes are shown here. There are these big molecules of double-stranded DNA supercoiled around proteins. So they're all clumped up and spun together to make up these big chromosomes. And sexually reproducing organisms usually have 2 copies of each chromosome, so they're diploid. And the genes are on the chromosomes, which means the diploid organisms have 2 copies of each gene. And these genes are in individuals, and we have a population, which is a group of interbreeding individuals and their offspring. And a gene pool is all the genes and alleles with the gametes produced by adults in a population. So an allele is a type of gene, basically. And so the gene pool just has all the different genes that are in the population. If an individual doesn't reproduce or doesn't survive to reproduce, their gene isn't part of the gene pool because it doesn't get passed on to the next generation. So for it to really be in the gene pool, it has to get passed on to the next generation. And when we talk about genetic diversity, we can look at it at different levels. So we can think about how many different genes there are in an individual. We can look at the genes on chromosomes, and so we get 1 copy from each parent. We can look at the diversity of genes in an individual. So do they tend to have the same copies of different genes, or do they have different copies? And then we can look at within a population, what's the variability of genes when we look at the group of organisms that are interbreeding. And then we can also compare genetic variability among different populations. So in this case, they're showing these mountain goats as living on different mountains, and so we can look at the genetic variation on these different mountains and compare it. So there's 3 levels of genetic diversity within a species. Within an individual, and we usually look at something called heterozygosity. And if you're heterozygous for a gene, then you have 2 different versions of that gene, 2 different alleles in your genetic code. And if we look at that across lots of genes, then we can get the proportion of genes where we have more than 1 copy of that gene. We can look at among individuals within a population, so how much variability is there among different individuals, and then we can look at among populations within a species. But a lot of this has to do with how many copies of a gene there are, and this heterozygosity is an important measure. So with genetic diversity, we're looking at the number and frequency of different genes. New genes come from mutations and the movement of individuals from another population. Movement may occur frequently, and mutations that lead to successful new traits are rare. So these mutations happen all the time, and they're just an error in the code, usually like a copying error. So every time you make a new cell, every time new eggs and sperm are made, all those genes have to be copied. And sometimes there are errors, and those are mutations. Most mutations are not helpful. Making an error in the code is often bad. A lot of miscarriages, they think, come from mutations that are not helpful, and you can have a miscarriage because your body's trying to get rid of, or the embryo just didn't survive because of this mutation. Occasionally, mutations are helpful, but the rate at which new helpful mutations occur is very low. So this doesn't happen very often, but it does happen sometimes. That's how we get new variation. But it takes a long time to get some new good genes in a population. Much more frequently, new genes will come from an individual dispersing from one population to another. So if there's a set of genes in one population and a different set of genes in a second population, if an individual moves and carries some of those genes from one to the other, that can introduce new genes into a population. But because this mutation rate is very low, we don't want to lose genetic diversity, especially if it's unique to a population. And that's why it's important to preserve genetic diversity that's already there, because these mutations just don't often lead to helpful new alleles. It happens occasionally, but not fast enough that we should just allow genetic diversity to be eliminated from different species.